Today we're diving into the world of structural genomics. We're going to figure out what it is, how it's done, and why it's pretty much one of the most important quests in biology right now. A mission to read the blueprint of life, but in 3D. So think about it like this. The Human Genome Project. It gave us the entire book of life. Every single genetic word all spelled out for us. But here's the huge catch. Just knowing the words doesn't mean you understand the story. The main characters in that story, proteins, they only really show you what they do based on their 3D shape. Without that, we're just staring at a long list of letters. It's not the epic of life, it's just data. And that's where structural genomics swoops in. The mission? To create a complete 3D atlas of all these molecules of life. The goal is to take that massive library of gene sequences and translate it into the actual physical shapes of proteins, so we can finally see how they work.
Okay, so let's start with this central problem, all those unread chapters in the book of life.
You see, a gene sequence is kind of like the recipe, but the protein's 3D structure, that's the finished dish. And here's the really crucial part. Over millions of years of evolution, a protein's physical shape, its fold, has been saved and passed down way more reliably than its genetic code. This makes the structure an incredibly powerful clue to figuring out what a protein actually does, even when its genetic sequence has mutated beyond recognition.
Understanding this is one thing, but actually mapping it all, well, that's a massive undertaking. It requires a truly global effort.
And this isn't some small, isolated project. No way. It's a huge, coordinated, international collaboration. You've got massive initiatives like the NIH's Protein Structure Initiative in the US, the Protein Structure Factory over in Europe, and Japan's Protein 3000 project. They're all working together toward this one huge goal of mapping the entire protein universe.
Now, these efforts are really being pushed forward by two different but complementary forces. On one side, you have publicly funded groups like the NIH. Their goal is broad coverage. They want to build a complete library of every possible protein shape for all scientists to use. On the other side, you've got the private sector, and they're way more targeted. They're focusing on proteins that are medically important, the ones that could become the next blockbuster drug. And you know what? Both of these approaches are absolutely vital to push the science forward.
So, how do they actually do it? Well, you should picture less of a quiet, pristine lab and more of a high-tech factory, with this really complex assembly line designed to turn a single gene into a 3D model.
This whole process is a pretty intense pipeline. It starts with just picking and cloning a target gene. Then, the scientists have to basically coax cells into making or expressing that protein in really large amounts. After that, the protein has to be purified and prepped for analysis, and only then can they collect the data to calculate that final 3D structure. It's a high-stakes process where at any one of these steps things can, and very often do, go wrong.
And the single biggest challenge, the great bottleneck in this whole factory, is crystallization. See, to use the most common method, X-ray crystallography, you need a perfect, beautiful little crystal of your protein. It's like getting all the molecules to line up in a perfect grid so you can bounce X-rays off them. And this is just notoriously hard and unpredictable. It can take months of trial and error, and for a lot of proteins, it might just never happen at all. Scientists basically have two main tools in their toolbox for this. X-ray crystallography is the workhorse. If you can get a good crystal, you can solve a structure super fast. But as we just said, getting that crystal is the real challenge. The alternative is something called NMR spectroscopy. Its big advantage? No crystals needed, but it's usually limited to smaller proteins, and collecting the data can take months.
The reality of this whole process is a funnel, a really steep one. For every 100 protein targets that go into the pipeline, maybe about 2/3 even get successfully expressed. From there, the numbers just plummet. Only about a quarter of the original group make it through purification. And by the very end, you might be left with just 5 successful structures that get deposited in the public database. It's a brutal numbers game. But for those 5 that actually make it all the way through, the payoff is absolutely immense. And this is the so what part of it all. How knowing a protein's shape actually leads to real-world breakthroughs.
Because here's the crucial point. Structure reveals function, and that knowledge saves lives. By seeing the 3D shape of a key receptor, scientists can design better drugs for type 2 diabetes. By mapping out a viral enzyme, they can create inhibitors to fight hepatitis C. And by understanding the exact shape of the tetanus toxin, they can figure out how to block it. These are direct, life-saving applications.
Structure also tells us these incredible secrets about evolution. A classic example is the first two protein structures ever solved, myoglobin and hemoglobin. If you just look at their gene sequences, they seem pretty different. But the moment scientists saw their 3D folds, bam! It was obvious they were related. They clearly shared a common ancestor. The structure tells a story that the sequence just can't.
Okay, so when a new structure is finally solved, it usually leads to one of 4 different outcomes. The first one is the simplest case. You get a familiar shape, and it has a known function. This is still really valuable because it helps us see how evolution has made all these little tweaks to a common molecular design for tons of different jobs across the tree of life.
The second outcome is way more exciting. You get a familiar shape, but for a protein with a total mystery function. The shape itself gives you a huge clue. If it looks like an enzyme that cuts DNA, for instance, you can guess that your mystery protein might do something similar. It gives scientists a clear place to start their experiments. It's like finding a tool you recognize, but in a completely new workshop.
Now the third possibility is fascinating. You get a completely new shape, but for a function you already know. This shows us that nature has found multiple different ways to solve the exact same problem. It's a beautiful example of convergent evolution at the molecular level, proving there's more than one way to build a biological machine for the same task.
And finally, the fourth outcome. This is the Holy Grail. A totally new shape with a completely unknown function. This is pure discovery. It opens up an entirely new branch of biology, showing us mechanisms we never even knew existed. This is the biological equivalent of a mapmaker discovering a whole new continent.
And all of this brings us to the future. With thousands of structures already solved and the technology getting faster and cheaper all the time, where is this all headed?
Well, the next frontier is something called structural chemogenomics. It's a big phrase, I know, but the idea behind it is even bigger. It's all about designing selective small molecules. Think of them like tiny keys that fit specific protein locks. But instead of designing a drug for one target at a time, we can use the entire 3D library of proteins to predict how one key might fit hundreds of different locks all over the body. That could lead to much safer and much more effective medicines.
And the impact here goes way beyond human medicine. This kind of structural knowledge can help us create targeted pesticides for agriculture, design more efficient enzymes for industrial manufacturing, and give protein engineers the tools they need to build brand new molecules from the ground up.
So we're left with this thought. The ultimate goal of all of this is a complete 3D atlas of life. Once we have that map, once we can finally read every single chapter in the book of life, the potential is just limitless. The question then isn't what we can solve, but what problems we'll choose to solve next.